So I'll do this in English so everyone understands what I'm talking about.
There you go.
Okay. So I'm going to talk about three areas today. First is our application of this technology in dendritic cell therapies, in T-cell therapies, and also in stem cell therapies for regenerative medicine. And our mission is really two missions. One is to increase the potency of cell-based therapeutics, because we believe that one of the biggest challenges in this field is to invest in a program for phase 2 or phase 3 and come out with an ambiguous therapeutic result. The second area that we're focused on is in decreasing the complexity of cellular-based therapies. So MaxCyte is located outside of Washington, D.C. We're a cash flow positive company, been in business for about 12 years, venture-backed. And the technology, the basics technology, is it's an automated sterile electroporation system where we can process 100 milliliters of cells in about 20 minutes, again in a sterile closed system, FDA cleared for a number of clinical trials. We use this technology in a number of different markets. We work in drug discovery, where we sell this technology to big pharma, and into biologics development and manufacturing. I won't talk about that today because we want to focus in on cancer immunotherapy. Our internal expertise is really around modifying primary human cells with this non-viral electroporation method. If you use electroporation in your grad school, this is very different. This is a technology that's very efficient. We can load basically 95% of the cells that we process, and the viabilities are 95% plus. So it's a very gentle, very rapid, and again sterile technology. We have pioneering patents in this area, and the original patent was licensed from Harvard Center for Blood Research when we formed the company. In parts of our business, we have 15 of the top 20 pharmaceutical companies have acquired this technology for drug discovery and biologics development, and we also have a number of partners in the cellular therapy space. The first thing I want to talk about is that we out-license this technology to companies who are developing cell-based therapies. And I want to talk about two of those. First is in dendritic cells, and the second in endothelial precursor cells. You've seen this schematic in a bunch of different ways, but basically you're taking apheresis from the patients. You then mature dendritic cells. In our case, we're loading mature dendritic cells. You can either co-culture those cells, but in our technology, we actually load the antigen directly into the dendritic cell using our technology. We do it in a large volume, so you can process the cells one time, aliquot the cells, freeze them down, and then give subsequent doses to the patient. So the patient really only has to be their cells processed one time. They come back to the center to get reinfused. What we're doing here in terms of increasing potency, we can process, basically load any molecule in any cell. So we can load DNA, RNA, small molecules, and protein, and that's important in this application where we're taking the dendritic cells, we're loading them with a lysate from the resected tumor. So we take the tumor from the patient, we lyse it down, and then we take the protein and we load it into the cell. It goes through the class 1 pathway, which we believe increases the potency of the cellular product. I'm not going to go into the details, but we're showing in vitro about a 15 to 20 increase in potency of this, and we're in the clinic right now with our first partner, which is Medinet in Japan, who use this technology in 5 cell processing centers all through Japan. It's considered a practice of medicine in Japan because you're taking the patient's own peripheral blood mononuclear cells, you're taking the patient's own dendritic cells, and you're combining the two. So we're beginning to get information and clinical data from these experiences. It's not a controlled clinical study, so we're trying to understand more and more how the in vitro data translates into human data. We also have just recently licensed the technology to a large company, biotech company in India, who will actually be doing a full-scale clinical trial in glioblastoma using this technology. Lastly, we have full rights to this outside of those 2 territories, and we're currently working at Duke to create some preclinical work that will lead into a company-sponsored IND for this dendritic cell therapy. The second area I want to talk about is more very regenerative medicine. We consider dendritic cells actually regenerative medicine because you're really regenerating the immune system for patients. But here we're working with United Therapeutics in the treatment of pulmonary arterial hypertension. And as you know, this is a disease where the blood dumps out into the lung. What happens in pulmonary arterial hypertension is that the arterioles become narrow, and the microvessels, you lose microvessels, and there's a backup in the heart, and it's a devastating disease. So what we're working with United, public company and outside of Washington, D.C., is an engineered endothelial precursor cell that overexpresses eNOS. So what we're doing is we're putting a plasmid, nitric oxide synthase, into the cell, and then putting those cells back into the patient. And some of the preclinical work we've done is we have a model here where you can show this was, we induced, well, we didn't, but United Therapeutics induced pulmonary hypertension with an agent monocrotaline. The systolic pressure went up from about 18 to about the mid-40s. This was cells that were endothelial precursor cells that were not engineered using our technology. And in this case, these cells were given about 3 weeks out, and you see a rather dramatic reduction in the systolic pressure of these rats in this case. Then in this small series of panels here, these are rat lungs that have been perfused with fluorescent microparticles. And this is a normal rat lung. This is one that has been treated with monocrotaline. You see that narrowing in the arterioles. And this is obviously a different rat, but this is the rat that had been given monocrotaline first, and then the engineered cells afterwards, and you see a regeneration of those arterioles. We think that's indicative of what could be happening with these patients. There's also an overall survival benefit that supported the phase 1, phase 2A trial. So there was a phase 1, phase 2 trial. We didn't, but United dosed about 12 patients. There was no dose-limiting toxicity. There was a rather profound drop in right ventricular systolic pressure in these patients. And there was a 6-minute walk where we were able to show preliminary efficacy. And although we can't talk about the plans, that's up to United to do that, there's a multi-site phase 2B that we're hoping will take place in 2012. Let me move a little bit to another strategy that we've been doing is seeding this technology into academic centers where we provide clinical non-commercial licenses to a handful of groups that are doing, we think, some really interesting work. And then once that proof of concept is given, we have the right, frankly, to sit at the table and work with the university to find commercial partners, and that's what we're doing in a couple of cases. The first case I want to talk about is what our friends at Bluebird just talked about, which was a very interesting T-cell therapy using lentiviruses. And this was two studies that were co-published at New England Journal and in Science, where they had treated 3 patients. Two of the 3 patients with lentivirus had complete remissions, and the third patient had a partial remission. This is a very interesting chimeric antigen receptor. There's an Fc on the outside of the cell that can be engineered for any receptor, and an internal T cell signaling modality. What's interesting about this is it's really independent of the surface concentration of the target cell antigen, and it's also really independent of the immune status of the patient. What we're doing here is working with Carl. The issue with the issue with lentivirus isn't the toxicity we talked about earlier. The problem is that because it's such a strong effect, any cell that has CD19 is going to wipe out. And what Carl was able to show, Dr. June was able to show, is that one T cell could take out about 1,000 cancer cells. And in this case, the concern is that if you have an antigen that would present on a normal cell, you're going to wipe out those normal cells as well. CD19 is a special case. So we've been working with Dr. June to turn this into an RNA-based therapy, where we're loading RNA in the cell that expresses the CAR. It has a transient effect, and again, we can do large volumes. So you process the cells one time, which is what we're doing here. So this is the lentivirus process. It takes about 12 to 15 days we've seen this in several other presentations, where you send it, this is a little airplane or a truck, you send it to a centralized facility where they do the processing. With our technology, it's the same process, although you cut the time down by about 3. Now, what's really interesting and what we're focused in on, because our technology comes from the transfusion medicine world, and because we can do large volumes and we do it very efficiently, our focus now is on actually not going through maturing dendritic cells or T cells, but actually processing cells directly from apheresis. So you take the patient, you apherese the patient. There's over 3,000 apheresis instruments around the world right now. And we have data that shows that you can get a similar sort of response if you take our technology, load RNA into a gamete cells, a full peripheral blood mononuclear cell population, and you can do that within a few hours, do it close to the patient, reduces the complexity dramatically, also reduces the cost, and we think could be a real major difference in how cell therapies will be delivered in the future. So with that, I'll stop, and thank you very much.